In this video, we're going to talk about the nervous system, and specifically, we're going to talk about what neurons are and how they work through action potentials. First, we're going to talk about the parts of the nervous system. The nervous system is broken down into the peripheral and the central nervous system. The central nervous system is the brain and the spinal cord, and the peripheral is going to be everything else. The peripheral is further broken down into two parts, autonomic peripheral nervous system and the somatic peripheral nervous system. And the somatic peripheral nervous system is going to control the voluntary muscle movement. And then the autonomic peripheral nervous system is going to control the involuntary muscle movements. And then that is further broken down into the sympathetic and parasympathetic. Sympathetic is arousing, or you may have associated that with something called the flight or fight response. And then parasympathetic opposes the sympathetic, which means it's more of a calming, or that's responsible for the calming feeling. So when we're talking about the nervous system, the neuron is the basic structure of the nervous system. This is the structure that receives electrical signals, transduces that electrical signal down its axon, and passes it from neuron to neuron. And so there are a couple of parts of a neuron you should be familiar with. The first is the structure here that we call the cell body. And the cell body has several dendrites here. That prefix dendr means tree, and it looks like little tree structures. These are the structures that receive messages from other cells. So a chemical message is received, and it's passed down this long tail-like structure, which we call an axon. And the axon just passes the message away from the cell body, and it always moves away from the cell body. And that's because there are these little blank spaces in between these cells that we call nodes of Ranvier. They ensure that the signal only goes in one direction. These structures here, these yellow oval structures, are myelin sheath, and these myelin sheath is a coating around the axon, and they're made up of something we call Schwann cells. So the myelin sheath is made up of Schwann cells wrapped around the axon, and they serve the purpose to enhance that electrical signal being pushed down the axon. So they enhance that transduction of the chemical signal. And then this axon is going to go to what we call these terminal buttons, which will then interact with dendrites or multiple dendrites of other neurons. There are three types of neurons: a sensory neuron, which is going to receive an external stimulus; an interneuron, or also known as an association neuron. This is going to mostly be involved in sending messages to the brain for processing; and then there's a motor neuron. So an example of a reflex that requires these neurons is the knee-jerk reflex. So you tap your knee, and a sensory neuron senses that external stimulus, and it's going to go straight to a motor neuron, meaning that's what causes your leg to kick. However, more advanced external stimuli would go from a sensory to an interneuron, which is the intermediate, which is then passed to the motor neuron to cause some function or some response. There are a couple terms we have to talk about when we get into, before we actually start talking about what an action potential is. So the first term that you need to be familiar with is what a membrane potential is. The membrane potential of a neuron is just the difference in electrical charge across the membrane. So that's talking about inside versus outside of that neuron. At resting potential, or at its resting state, the membrane potential is about -70 millivolts. That's what MV stands for. And it's maintained at this negative value thanks to this sodium-potassium pump, which we talked about very early in the year, but we're going to talk more in detail now. So there are gated ion channels. Remember when we talked about cell communication, gated ion channels. And there's two that allow this action potential to occur. So when a stimulus is received or a neuron is excited, the sodium-potassium pump is stimulated to open. What's going to happen is sodium is going to move into the cell. And sodium is always found in higher concentrations outside of the cell. So if we open up these sodium channels, sodium is going to rush down its concentration gradient into the cell, causing the inside of the cell to become more positive because sodium is a positive ion. So we call that depolarization, becoming more positive. Now, potassium is always found in higher concentrations inside of the cell. Again, this is due to the sodium-potassium pump. So what's going to happen if we open up a a potassium pump? It's going to move down its concentration gradient. It's going to go from inside the cell outside of the cell, making the inside of the cell more negative because we're losing a positive charge. Becoming more negative is something we call hyperpolarization. So thanks to the sodium-potassium pump, which maintains our resting potential, and then these other gated ion channels, these sodium and potassium pumps, we can actually cause what's called an action potential. And so the movement of sodium and potassium causes this wave of depolarization. And essentially what happens is that axon is stimulated over the resting potential or the threshold potential. Once a threshold is reached, we can now exhibit an action potential which causes an electrical signal to be passed down neuron to neuron. So here's a pretty basic graph that you should be able to interpret and redraw if I asked you to. So what you can see is time in milliseconds and voltage. So remember that at resting potential, the neurons are at negative 70 millivolts thanks to the sodium-potassium pump. Well, there is some stimulus that's going to cause the sodium channels to open. Remember, that's going to cause sodium to rush into the cell, causing it to become more positive. And remember, we call that depolarization. So we have this depolarization event. We're now at positive 40 millivolts. Once that action potential is reached, we're then going to open up the potassium channels. Potassium channels are going to allow what to happen? Right, potassium is going to move out of the cell, causing the cell to become more negative. We call that depolarization, hyperpolarization or repolarization. So we start seeing a big drop. And what we have here is a refractory period. This refractory period is just referring to a time where we actually overshoot that resting potential, but eventually it's going to come right back up to our resting state at negative 70 millivolts. This is an action potential, and this always moves down the axon in one direction. So talking a little bit more about the sodium-potassium pump, which is maintaining the negative 70, what happens with the sodium-potassium pump is that sodium is always going to be pumped outside of the cell. 3 sodium are pumped outside of the cell, and 2 potassium are pumped inside of the cell. This is going to maintain that the inside of the cell is always more negative than the outside because the outside has more positive charges. And so this is an example of active transport because it uses energy to pump sodium against its concentration gradient. So it's going to continuously pump 3 sodium out of the cell and 2 potassium inside of the cell. And then when those gated ion channels open, we'll have potassium and sodium flowing down their concentration gradients, leading to these action potentials that we're talking about. So also associated with neurons are synapses. And there's a couple of terms that we'll need to talk about. So the presynaptic neuron, so the neuron that is receiving the signal and then passing it down, is going to contain vesicles with neurotransmitters. So that action potential has continuously moved down the axon. We start getting to those, to the very terminal buds of the presynaptic neuron, and there are vesicles with neurotransmitters. An example of a neurotransmitter is acetylcholine. Depolarization, so remember, what is depolarization? It's going to be making something more positive. So depolarization causes an influx of calcium. That influx of calcium causes vesicles to fuse with the membrane, and then they secrete their neurotransmitters into something we call the synaptic cleft. That is going to be the gap in between 2 neurons, and that occurs via exocytosis. Those neurotransmitters are going to bind with the receptors on the postsynaptic neuron. The postsynaptic neuron is the neuron receiving the signal. And that postsynaptic neuron will either be stimulatory, so cause another response, or inhibitory. It just depends on the receptors found on it. And then any neurotransmitters that are left in the synaptic cleft are either retaken up by the presynaptic neuron, or they're broken down by something called esterases. So now to give you an illustrative example of how the human body is able to receive an external signal, pass that signal to the brain, and then cause a response. So if we're talking about eyes and light. So here's the basic structure of the eye. Don't worry about knowing the different parts of the eye. So photons, which are those excited light particles, are going to pass through the lens, and that is going to then reflect onto the retina back here. In the retina, we have these cones and rods, which are photoreceptors. So they're going to be receptors that are receiving these photons. And this activates a pigment called rhodopsin. So rhodopsin is now activated. If you remember from cell communication, we talked about G protein signaling molecules, or G protein coupled receptors. We're going to activate that, which leads to a series of cell responses. We're going to close sodium channels, so this is going to cause hyperpolarization. And then this is going to lead to impulse being sent to the optic nerve, and then that optic nerve is going to run to the brain for processing, allowing us to now see light. So this is an example that you should be able to walk me through of how the eye can receive that light external signal and pass it to the brain for processing.